Hi, and welcome back to the wonderful world of the scientific hypothesis. My name is Brad Alger, and in this short video, we'll dig a bit deeper into the huge topic of reproducibility in science, specifically into the difficulties in which workers in one laboratory have when trying to repeat observations that are made by workers in another laboratory. I'll highlight a few factors that can hinder direct reproducibility, which you'll remember is the attempt to duplicate previous findings exactly. The examples are drawn from the biological or biomedical sciences, but similar concerns affect all of science. I'll first illustrate a few of the common sorts of problems that can arise when the materials that scientists use are variable or unreliable. I'll then discuss a striking example that shows how the extreme subtlety of nature can really mess up even the most conscientious quest for reproducible data.
So let's look at some of the external barriers that can hinder the ability of one lab to reproduce the findings in another one. There are two major classes that I'll talk about. The first is untrustworthy materials, and the second is a group of difficulties that I'll lump together under the title of complexity of nature, and I'll go into one specific example in some detail. Reproducibility is one of the core scientific values. Science seeks true explanations or hypotheses, and reproducibility of results is one sign that we're on the right track to the right explanation. Reproducibility is also the basis of claims of scientific objectivity. Scientific observers have to agree with one another, and they can't very well do that if the results disagree. But reproducibility alone does not guarantee that we're on the right track, and irreproducibility doesn't imply that we're on the wrong track. Nature is very complex, and reproducibility can be very hard to attain. Now, in this video, I'm going to focus on these latter 2 points and try to address the questions of why reproducibility fails. What can go wrong? Most scientists rely on materials to do their experiments, especially in the biological sciences. And one common strategy is to use cell lines. These are cells that have been maintained for long periods of time and are often available from commercial suppliers or colleagues. Obviously, the cells have to be correctly identified. Cells from one part of the body, such as liver cells, are certainly not the same as cells from another part of the body, such as spleen cells, and misidentification could lead to a number of problems. However, the difficulties with cell lines are much more extensive and subtle than this. Cells are grown in dishes and maintained in laboratories. The cells are strongly affected by the particular fluid, the growth medium that they grow in. Does it contain serum? If so, what kind? What is the batch number? What is the amount? And all of these things can have effects. What are the lighting conditions in the lab? What are the materials of the dishes made of? Different kinds of plastics can leach different kinds of chemicals into the medium and affect the cells. What are the fluid levels of the cells? What are the age of the cells and the origin of the cells? Is there any contamination present? These are just some of the kinds of factors that can affect the properties of cells that are studied. So even though another laboratory might seem to have the same type of cells, the cells themselves might in fact be extremely different. Another common type of strategy employed by biologists is to use antibodies. These are molecules which are designed to be specific, that is, to recognize and to bind to or to stick to only certain kinds of proteins. This is their great strength, and so certain proteins can be identified and their functions studied by use of antibodies which will adhere to them and potentially interfere with their function. But what if the antibodies are wrong? What if they're misdirected? What if you had an antibody which is supposed to be recognizing a sodium channel, which is a type of protein in cellular membranes, and it's really sticking to calcium channels, which is an entirely different type of protein? If you have these antibodies which are misidentified, then you'll get the wrong results, and moreover, other investigators using genuine sodium channel antibodies will not be able to reproduce your results. Problems with untrustworthy materials can be very serious. However, in principle, they should be relatively straightforwardly fixable. In a sense, many of them involve quality control problems, and therefore we can try to improve the production of these materials. The end user, that is the scientist actually using these different materials, should engage in checking the quality of them or the identity and so on. But this is not always practical. Not all laboratories have the ability to do thorough genetic analyses and so on. One strategy, which is not always successful, but which should always be tried, is to maintain absolutely constant conditions in the laboratory so the variability there is reduced. And finally, control studies in which a given result is checked using entirely different methods and different approaches and different materials can be very effective in combating the problem with untrustworthy materials. Problems associated with the complexity of nature are of an entirely different kind and may cause far more serious problems. We often don't know what it is that we don't know, and therefore we can't take it into account. If relevant variables are among the things that we don't know about, then we can't report all the relevant variables that affected our experiments. And variables that we thought were unimportant may in fact have major influences. And yet the failure to account for minor factors may prevent reproducibility in the hands of other researchers. To illustrate this, I want to consider one dramatic example that's been published. I call this the case of the stressed out lab rats, and the reference for this material is given at the bottom of the slide. The general idea, and this is an overview, is that this was a large laboratory and they were using behavioral techniques to study laboratory rats. Now, the laboratory consisted of both females, graduate students, postdocs, and technicians, as well as male graduate students, postdocs, and technicians. And they noticed a very remarkable thing. Whenever the females did the experiments, then the results showed that the rats did not experience stress. However, when the males did the identical experiments using the identical equipment in the identical groups of rats, they concluded that, in fact, the experiments did cause the animals to experience stress. In other words, the two groups got entirely different and conflicting sets of data, despite the fact that everything seemed to be as well controlled as it possibly could be. So this was clearly of great concern and interest. What could be going on? The first thing they did to rule out the possibility that individual personality traits or other things had anything to do with the results by getting brand new identical t-shirts, and they gave one to each of the lab members and asked that person to wear it for an entire day. What they found was that the t-shirts themselves had the same effect as the entire person. So t-shirts that were worn by the female lab members did not cause the rats to experience stress, whereas placing the t-shirts worn by male lab members near the rats did cause them to experience stress. And after a long and very careful series of experiments, this is what they concluded: that males secreted chemicals, pheromones, and so on, and the rats detected these secretions, and this caused the rats to experience a stress response. This clearly was a remarkable study in a number of ways. You can easily imagine some other laboratories consisting only of females and other laboratories consisting only of males, and their results would continually conflict. Neither one would be able to reproduce the results found by the other one. So, in summary, these are some kinds of factors that support the conclusion that irreproducibility doesn't always mean that somebody did something wrong. Nature, especially in the biological, biomedical, and social sciences, is amazingly complicated. Some problems are caused by untrustworthy materials. Many of these should be relatively easily resolvable. Some problems may be much more difficult, as they will result from our ignorance of what relevant variables within the complexity of nature really are and how we can go about taking them into account. Nevertheless, because all of these things ultimately do deal with empirical matters, we should, at least in principle, be able to get a handle on what they are and do something about them. In the next video, I'll consider a number of the intangible factors that can go into influencing whether results are reproducible or not, and as you'll see, these may be much more challenging. Thanks for watching. Remember to give it a thumbs up if you like it and subscribe to hear more. See you next time.